So today we're covering chapters 9 and 11 in the lab manual, and uh, it does look like they're long chapters. However, we traditionally don't do the entire chapters. So we're going to be skipping a lot of things, so make sure that you keep good notes on what we're skipping. I'll show you exactly what we're not doing. And actually, I think it might turn out to be a short session. Okay. So this lab is mostly a vocabulary lab. You're going to be presented with quite a number of things that you should remember. Nothing you haven't seen already from lecture, or even from yesterday in some cases. We'll start off with what is an allele. All right. So remember, yesterday we were talking about individual organisms inheriting two sets of chromosomes, one set from each parent. Well, each set of the chromosomes has the same types of genes on them, right? But not necessarily identical genes. So there could be a location, a locus. Let me give you that one, which I forgot to write. Locus. This is the location. Oops. Locus. Location of a gene, or even a few genes, on a chromosome, or just the DNA. Okay, so for any particular locus on homologous chromosomes, they would have the same gene in that location. All right. So, for example, if you had this chromosome from one parent and this chromosome, this is before meiosis happened, so they don't look like tetrads, and they don't look like replicated chromosomes. The gene for, let's say it's a lady beetle, the gene for the number of spots on the lady beetle would be, I'm just making stuff up here, it would be located at this location, this locus. Guess what? On the homologous chromosome, it would be at the same exact locus. Okay, that's why crossing over works, because after crossing over has occurred, nothing's missing, because all the genes are located in the same places. All right. Now, this version of the gene, this gene and that gene may be coding for the same trait, but they may be different traits. I'm sorry, not different traits, but different versions of that trait. Alternate forms of the same gene. And that's what we call alleles. So maybe for this chromosome on the lady beetle, this one says no spots. Forget the spots. You don't need spots to survive. But this, so that's the first one here. This one over here. But for this other chromosome, the red one from the other parent, it says 11 spots. All right, and honestly, I don't know which one is dominant or recessive, but let's say the dominant one is the one with the spots. Well, this allele over here is going to be the one coding for the phenotype, for the actual physical expression. All right, if this is the dominant one. The dominant one always is the one that gets expressed physically. All right, so you have, now you understand, I hope, what an allele is. Allele is just an alternate form of a gene. I'll give another example here. For example, your earlobes. You can check this out now as you're listening to this video. Either you have attached earlobes like this, so you have no earlobe here that's hanging independently. It's just directly attached to your face, or you have a hanging earlobe. Okay, so one version of the gene codes for the unattached version, and the other version codes for the attached version. Okay. So that's a simple example of how alleles affect your physical appearance in humans. Right. So having two chromosomes, two sets of chromosomes with the same types of genes, remember that we call that being diploid, right? 2N diploid, right? Because diploid animals have two sets of chromosomes, you can carry the two genes that you carry, the two alleles, can be the same or they can be different, right? So they don't always have to be different. You can even have, in some cases, that particular gene is missing, so you don't have an allele for that. For example, the ABO blood group. O blood type means you don't have the A or B alleles. So I have to get you to understand what a genotype is and what a phenotype is. Right? So in terms, now that you understand what an allele is, I can explain genotype. Genotype is the actual genetic makeup of that individual in terms of that particular gene or trait that we're thinking about. Could be more than one trait. All right, so the genotype is not what is the DNA makeup of a particular individual. No, we're just concerning ourselves with one or two traits or three traits, whatever it is that you're researching. For example, what is the genotype for this condition with the earlobes? Are you recessive and have attached earlobes? So in that case, you have two copies of the recessive allele, or do you have two different copies, or do you have two of the same dominant alleles? So what is your genotype? Is it going to be big E, or big E, little e, both of which code for unattached earlobes? This is E. Each letter represents one of the alleles from each one of your homologous chromosomes. And in this case over here, you have little e, the recessive form. Okay, so what is the genotype going to look like? Is the genotype going to be homozygous? Let's say homozygous, for example, big E, or little e. Or is it going to be heterozygous? Two copies of the gene are different. We have big E and little e. And compare that to what a phenotype is. A phenotype is the physical expression. I've already said this before, but now we're going to clearly define it. Phenotype is the physical or outward expression that is caused by the genotype. Right. Now, there are some other factors. There are environmental factors. There's also the issue of epigenetics, but we're going to keep it simple here in Bio 11. All right, sorry, forgot which course we were teaching. Bio 11. We're going to keep it very simple, and we're just going to ignore those other forces. We're not going to talk about the environment affecting the expression of the genotypes, or other things like epigenetics. So just make sure you understand the genotype is the actual genetic expression, it's found in your DNA, and the phenotype is what you look like on the outside. The phenotype doesn't just mean what's your hair color or eye color or the color of your plumage if you're a bird or the number of spots on a lady beetle. It also means your behavior. What is it? How is it that you're behaving? So remember, genetics code not just for physical characteristics, but also for behavior. Okay, so there's another term that you have to be familiarized with, and it's dominance versus recessiveness. Right, so the genotypes can be a combination of any of those. All right, since your two copies can be different, right? The phenotype will depend on whether or not you have copies of the dominant allele or not. All right, so the rule is if you have a dominant allele, even if it's just one, it gets expressed, and the recessive allele does not get expressed. Right? Recessive alleles, in order for them to be expressed, you will need two copies of them. In other words, the absence of the dominant. All right. Okay, so when you have a condition where both alleles that are in the genotype are the same and they're both dominant, you call that homozygous dominant. For example, big E, big E. And it's just convention that when we are talking about dominant alleles, we write them as capital letters. And then the opposite of that, the recessive allele for that particular gene, is written with the same letter but in lowercase. So for example, homozygous recessive, both alleles are the same, but they're both recessive. In this case, we write E, little e, for attached earlobes. And don't forget the heterozygous condition where both alleles are different. So we have big E and little e. All right, so in the case of the heterozygous here and the homozygous dominant condition, in this case and this top case here, the dominant physical characteristic of having unattached earlobes is expressed. And the only time that an individual will have attached earlobes is if it is homozygous recessive. So for those of you out there who have attached earlobes, congratulations to your genotype. All right. For the rest of us, we don't know for sure what the genotype is unless we do a little digging around in our family history. Okay. All right, so here's an example that I was giving you just a few seconds ago. Okay, and basically says the exact same thing I just said. All right, so you guys can read through this slowly. Okay. Okay. So knowing this information helps you do a little deductive work or predictions. So, knowing genotypes of the parents will help you figure out what the offspring might have if they're not born yet. All right. So, for example, and we can use this thing called the Punnett square. This is a Punnett square. I should have chosen a different color here. These are Punnett squares. You can use these Punnett squares to help you figure out what the possibilities are for the offspring. Okay, so the Punnett square basically works by segregating the possible alleles for each gamete. Okay, so for example, what am I talking about here? All right. Let's say you want to represent what the possible combinations for the gametes of the male are. So the sperm, we're telling you here that this male is homozygous dominant. That means the sperm can only carry, any sperm that he produces can only carry dominant allele. And the same thing for the female here. Her eggs can only carry the dominant allele. So of course, when you want to figure out what the offspring are, everybody's going to be the same genotype as the parents. So here in this case, we're going to do the same thing. Okay. In this case, we're telling you that the parents are heterozygous. So questions can be asked. What are going to be the possible genotypes of the offspring? Well, okay, if this particular type of sperm fertilizes this particular type of egg, then you get this possibility. Okay, if this particular type of sperm fertilizes, sorry about that, this particular type of fertilizes that type of egg, and you get this. This is the only combination you can have. If this particular type of sperm fertilizes this egg, fertilizes this egg, then you get this combination. It's kind of the same thing as this, right? If this egg is fertilized by that sperm, you get the same thing. So these two are the same. Anyway, always write the dominant allele first, regardless of which order they came in. And in the case of having a in the case of having this sperm here fertilizing this egg, then you get this combination. Okay, so here's another example. Okay, let's say, for example, you have, here's the female, here's the male. Okay, let's say, for example, you have a male with, not this one, but a recessive condition, and the female with the dominant condition. What are going to be the results? You can pause the video and do it yourselves and compare the answers. Okay, so the result would be everybody's going to be a heterozygote, right? What's the phenotype for all the offspring? The phenotype for all the offspring is going to be, they're going to have the dominant condition of unattached earlobes. Right, so this also shows the law of segregation, which is something that Gregor Mendel thought of when he was doing his experiment on pea plants. All right, if you haven't read that from your lecture, please do so. Each organism contains two alleles for each trait. We know that, we've established that. And the alleles segregate during the formation of gametes. I showed you this yesterday, right? Independent assortment happens. Each gamete, egg or sperm, that contains only one allele for each trait, right, then contains only one allele for each trait, which is why we list them separately. When fertilization occurs, the new organism has two alleles for each trait, one from each parent, and it can be random. Right? That's why we give each one of the gametes a chance to fertilize all the other gametes. Oh, and by the way, when we're talking about single trait, this is an example of a single trait cross. But this particular example, where you have two parents and both parents are what do you call it? Are heterozygous. When both parents are heterozygous, we have a special term for that, and it's called monohybrid cross. Monohybrid cross. Right? And the phenotypic ratio, that is, what is the ratio of dominant versus recessive phenotypes? Phenotypic ratio for monohybrid crosses are always 3:1. Right. So what do I mean by 3:1 ratio? Only one of the possible offspring will be recessive, and the other three are dominant. So there's a 3 dominant against 1 recessive. So remember this, okay? Monohybrid crosses produce this type of ratio, phenotypic ratio. Phenotypic ratio. Okay, so you can determine the probability of a phenotype in the offspring using Punnett square, which is what we were doing before. So again, 3 out of 4 will be dominant, and only 1 will be recessive. Okay. So this kind of harkens back to the first lab, your scientific method. All right. You have an idea of what the ratios, the phenotypic ratios of the next generation should be. All right. So this is hypothesis. The expected phenotypic ratios is your hypothesis. Okay, in a cross with 2 heterozygous parents here, this, by the way, is lowercase p, right? You hypothesize that in the next generation, 3 out of 4 phenotypes will be the same, and 1 out of 4 phenotype will be different. 3 out of the 4 will be dominant, will have the dominant phenotype, and 1 out of 4 will have the recessive. All right, in this case, P, capital P equals purple, and lowercase p is yellow. And this is in terms of corn kernels. So this is the next example in the book. All right. So here we're talking about corn kernels and what color the corn kernels are. Are they going to be purple or yellow? And then we're told that the yellow is recessive, and that the parents were heterozygotes, both. So this is, again, this is a monohybrid cross. So this is a monohybrid cross. So you have this expected phenotypic ratio of 3:1. But then you go out and you do your experiment, you collect the corn cobs for the offspring, from these parent plants, which are the offspring. Each corn kernel is the offspring of the parent corn plant. And you calculate what are the actual phenotypic ratios. What are the actual phenotypic ratios? Well, you have to go out there and you have to count how many of the corn kernels are purple and how many of them are yellow. Right? Okay. So let me just take a look at the lab manual so that we don't get too far into it. Okay, so we discussed We discussed this and that. Homozygous recessive, genotype, phenotype. We talked about what an allele is. Okay. So here they're talking about the one trait crosses that are, and one example of that is the monohybrid cross. Okay, when you both individuals are heterozygous. All right, so in the lab we actually don't do this experiment, but it's more of a thought experiment anyway, so we can cover it here. All right, what is the expected phenotypic ratio if the parents of these tobacco seedlings are heterozygous? All right, this is another monohybrid cross. All right, so what are going to be the gametes, the distribution of the alleles among the gametes? Okay, the female parent, this is her genotype. So one gamete, it's possible to have one gamete with this capital C, which means green, and another one with a lowercase c. And the male parent is going to be the same. Okay, so what are the possibilities? Well, we know what the possibilities are. We're going to get a 3-to-1 ratio, right? One is homozygous dominant. Two of them are going to be heterozygous, and one is going to be homozygous recessive. Right? Whenever we have a monohybrid test cross, we have this 3-to-1 ratio. Okay. So in the lab, though, we would be doing this experiment, and we're going to do a little bit of the same thing here now. All right. Okay, so we'll go back now to the PowerPoint. Okay, so here we have an example of that corn kernel, just like the one in the book. And again, capital P stands for purple, right? All these little corn kernels that are purple either have the dominant homozygous condition or the heterozygous condition. And these little p ones, the yellow ones, are all going to be recessive. So they have 2 copies of the little p. All right. So I actually took the liberty of just doing this for you guys last night. And what I did was I counted each purple corn kernel for these 3 rows that I've put a little blue rectangle around. Right? And what that shows me is in sample 1, which is row 1, this is row 1, row number 1, I have 16 purple corn kernels and 4 yellow corn kernels. And my phenotypic ratio, my actual phenotypic ratio for that particular row is 4-to-1. Basically, I just divide both numbers by 4. All right, and I get 4 and 1 over here. So this at 4 here and 1 over here, 4-to-1 ratio. Now, that's not 3-to-1. It's way off, actually. But you have to think we have a very small sample size, right? This is why another reason why large sample sizes are important. In fact, this particular experiment drives that concept home by having you do 3 rows, which I did here, and you guys can copy these numbers or do it, count it out yourselves from the picture that I present here in the PowerPoint. And you know, the closest one is this one in the middle. But as it turns out, if I add up all the 3 rows so that I increase my sample size, I get 48. You get 48 purple corn kernels and 16 yellow corn kernels. And if I divided both numbers by 16, I actually get an almost perfect 3-to-1 ratio. All right. And if we were in class, everyone would have a different corn kernel, and we would be able to obtain class data. So we would have all the purple corn kernels that were counted by everyone in class, and all the yellow corn kernels that were counted by everyone in class. And hopefully, we would also get a 3-to-1 ratio. But it doesn't always work out that way. All right, expected phenotypic ratios are what happens in controlled, perfect conditions. In the real world, where there are so many other variables affecting the situation, the ratios don't always end up as perfect as the actual phenotypic ratios don't always end up as perfect as the expected phenotypic ratios. All right, so in this case, you go out and you do some statistics. Are your actual phenotypic ratios close enough to the expected so that your hypothesis can hold, or are you going to go ahead and reject your hypothesis? All right, so you're going to need statistics. You don't want to eyeball it. You don't want to just say, oh, 3.4 is close enough. No, or 4-to-1 ratio is close enough. Now you want to make sure you put it through some rigorous mathematical testing so that you can tell, this is the purpose of statistics, and that way you can tell whether there's a significant difference between your actual phenotypic ratios and your expected. If so, if the actual significant difference, then you're going to have to go back and rethink your hypothesis. As it turns out, we don't do that because when we actually go out and do these experiments, we always get close enough to 3-to-1 ratios. Okay, I'm going to share my screen again so we can answer the question. All right. All right. So, this is the tobacco seedlings, which we're not going to do. All right. Do your results differ from the phenotypic ratios? You can say yes, expected. Right, expected phenotypic ratios are only possible in controlled conditions. So you'd have to control how many gametes were out there, how many pollen grains fertilized, how many egg cells in the ovaries of the plants. Excuse me. Was your class data closer to the expected phenotypic ratios? And the answer should be yes. Okay. And the reason for that is you have a larger sample size. I know it doesn't ask the reason, but we'll write it down anyway. So here we're doing the corn kernel. Same thing, okay? Now, here's my issue with this lab manual. This is incorrect, okay? So this is incorrect. We have Both parents are going to be the same. They're not going to be different. So this is going to be just like the tobacco seedlings. All right, big P, little p, big P, little p. All right, we have the same situation. Okay, so fix this. Big P, little p. Fix that in your book. What is the expected phenotypic ratio among the offspring? It should be, again, 3 to 1. All right. And this, see the PowerPoint, PPT for PowerPoint. So I have the answers for you there. Do your results differ from the expected phenotypic ratio? Our results, we'll say yes anyway. Our results, yes, they differed because, again, the same reasons as before. Because expected phenotypic ratios they only occur under perfect conditions. Right? They don't occur in real world conditions. Was your class data closer to the expected phenotypic ratio than your individual data? For most of you, it would have been yes. On occasion, some people would what do you call it? Some people would hit it right on the mark. But we're going to say yes anyway. Okay. Class data is closer to expected, right? It's closer to expected phenotypic ratios for the same reason that we said before, due to larger sample sizes. Right? You increase your sample size and you reduce the random effects of chance. Right? Just like if you're throwing pennies, right? If you just throw a penny 3 times, there's a big chance that you're going to get unexpected results. If you throw the penny 4 times, there's a large chance that all 4 times is going to give you tails or heads. But if you throw the penny 100 times, there's a closer chance, a better chance, that you're going to get half head and half tails as results. You're increasing your sample size. Okay, one trait genetic problems. Let's see if we can do this. In pea plants, purple flowers, right, capital P, is dominant, and white flowers, lowercase p, is recessive. What is the genotype of pure breeding white plants? Okay, so pure breeding means that they produce plants with only one phenotype, right? All the flowers on that plant are going to be white. Now, in order for the recessive condition to present itself as a phenotype, it must be present as homozygous as a homozygous condition. Right, so you have little p, little p. If pure breeding plants are crossed with these white plants, what phenotype is expected? If pure breeding purple plants, right? So that means we have homozygous dominant versus versus recessive, homozygous recessive. I made a mistake here. And in that case, everybody's going to be heterozygote, right? Huh? Yep. So big P, little p. Use a little Punnett square to figure it out. In pea plants, tall, capital T, is dominant, and short is represented by lowercase d, the recessive form. A heterozygous tall plant is crossed with a short plant. Okay. All right. So look, we know what the genotypes are going to be just from what they told us. A heterozygous tall plant, that means big T, little t, is crossed with a short plant, little t, little t. The only way the plant can be short is if it's a recessive, homozygous recessive. So it's little t, little t. So in this case, you get big T, and little t, and big T, and little t. Half will be heterozygotes, and half will be recessive. So you get a, you can have a 1-to-1 ratio. Unexpectedly to the farmer, two tall plants have some short offspring. What is the phenotype of the parent plants and the short offspring? Well, the short offspring are going to be little t, right? We established that you can only be short if you're homozygous recessive. Now, the only way you can homozygous recessive offspring is if you have parents that are heterozygotes. All right, big T, little t. Parents, if both parents are big T, there's no way that you can get the recessive form. Okay, in horses, two trotters, you learned a little bit about horses here. In horses, two trotters are mated to each other and produce only trotters. Two pacers are mated to each other and produce only pacers. So what is that telling you? It tells you that the parents are homozygous dominant, all right? Or at least one of the parents is homozygous dominant and the other one might be heterozygous. When one of these trotters is mated to one of the pacers, all the horses are trotters, for sure, okay? So all the trotters are producing trotters, even when they're mated with pacers. Create a key to show the cross. All right, so the trotters, let's do that. We know now that the trotters are dominant. So capital T. This is our key, trotter. And little t for pacers. Okay, so the cross will be, we know that they're definitely homozygous dominant because they all produce trotters even when they were mated to pacers, right? And pacers would be little t. So it's the same as this, okay? Except that the t's mean different things. Our brown dog is crossed with two different black dogs. The first cross produces only black dogs. And the second cross produces equal numbers of black and brown dogs. What is the genotype of the brown dog? Okay, the first cross produces only black dogs, and then the second cross with the other dog produces equal numbers of black and brown dogs. Okay, so the way, see, here you have, in this case, you have equal numbers of one phenotype versus the other. So we know that the brown dog is going to be recessive. Let's call it little b, little b. Actually, I think they might just be looking for the words recessive. Okay. The genotype of the brown dog is recessive, and the first black dog would be homozygous. Increase the size here. The second black dog would not be homozygous dominant. It would be heterozygous. Right? Half of the offspring turn out to be brown, and the other half turn out to be black. Just like over here, half of the offspring were trotters, and half of the offspring, these were trotters, and the other half were pacers, or tall versus short. It's the same thing. In pea plants, green pods, capital G, is dominant, and yellow pods, lowercase g, is recessive. When two pea plants with green pods are crossed, 25% of the offspring, this is important, have yellow pods. What is the genotype of all plants involved? Okay, you do Punnett square. All right, 25% are recessive. Little g, little g. That means this is a little g and that's a little g. When two pea plants with green pods, both have green pods. Therefore, we know that they have the dominant gene. Both of them. And we also know that both of them have the recessive gene, the recessive allele, excuse me. All right, so the resulting offspring will have this 3-to-1 ratio also. Plants with green pods will be heterozygous, and plants with yellow pods will be recessive, homozygous recessive. A breeder wants to know if a dog is homozygous black or heterozygous black. If the dog is heterozygous, which cross is more likely to produce a brown dog? Okay. Explain your answer. Now, if you don't see it right away, you can do that Punnett square. Well, if I do this cross, what are the chances that I'm going to get some brown dogs? Okay, this is going to be, these guys are going to be black, and these guys are going to be brown. Oops, sorry. On the other hand, what did I do? On the other hand, let's do another Punnett square over here. So now we're going to do this one. Over here. Big B, little b. You guys can, if you want to figure this out, you can do it the slow way, or you can do it in your head. Whatever. If you're comfortable enough doing it in your head, that's fine. Okay. In this case, like I said before, 3-to-1 ratio, right? Only a quarter of the offspring will be brown, but here half of the offspring will be brown. All right, so you have your answer. Big B by little b, little b. Big B, little b by little, because, another b there, because this cross will, right, a 1-to-1 ratio. In other words, half of the offspring will be brown. If the cross in problem 6 produces 220 plants, how many offspring have green pods and how many have yellow pods? All right, 25% of the offspring will have yellow pods. All right, so they're asking you to do some math there. So what's 25% of 220? Well, just write 25% of 220. If the part 2 is the offspring are tall and how many are short? Number 2. The 1-to-1 ratio is this one. Yeah. Okay. So that's a little bit easier to think about. How many offspring are tall and how many are short is going to be the same number, 110. Half, right? All right, so now that brings us to 2 trait crosses. And just going to talk about that. 2 trait crosses involve 2 pairs of alleles. So now we're talking about 2 different types of traits being considered at the same time. All right, so we use a dihybrid cross just like we use a monohybrid cross. Dihybrid cross meaning both individuals are heterozygous for both traits. And if we do that, our phenotypic ratios are always going to be 9-3 to 3-1. Right? Okay, so when we're doing a Punnett square, remember that you have in gametogenesis, you have independent assortment, right? The chromosomes that separated independently find themselves in random gametes, right? All the chromosomes that were inherited from one parent don't travel together. They reassort themselves independently in the gametes. So when we're doing a Punnett square for this dihybrid cross, we have to kind of do like well, I'll show you. Or show you instead of explaining it to you. We have to distribute the possible gametes, the possible alleles for each gamete. So, for example, okay, we have 2 trait crosses in the corn, different corn cob. And this corn cob will have purple versus yellow corn cob of corn kernels. And some of those corn kernels that are purple or yellow will either be smooth or wrinkled. And we're doing a dihybrid cross. In which case both parents are heterozygous. So what do we do? Okay. Let's say, wrong color. Okay. Let's say here's the male and here's the female. Okay. So they're heterozygous for both conditions. That means they have both alleles for purple and yellow, both alleles for smooth and wrinkled. And all gametes have to have one or the other, right? They have to have one of the alleles and they have to have one of the alleles for the texture. So, well, we the possible alleles for the 4 gametes. All right. So one of them is going to be is going to carry the dominant purple and the dominant smooth condition. One of them might be dominant purple but the wrinkly condition. One of them will have the yellow condition, but the recessive yellow condition, but the dominant wrinkly condition. Sorry, dominant smooth condition. And some of them will have the recessive condition for both alleles, for both traits. All right, so both parents are the same, so they end up with the same distribution of gametes. All right, so what we see here is that we have, a big P by big P, big S by big S, big P, here. Big P by big P gives you P. Big P. Big S by big S. Okay, and so on. You can do the combinations yourself. But what we end up with is 1, 2, 3, 4, 5, 6, 7, 8, 9 purple smooth. 9 out of 16 are purple and smooth. They have the dominant condition. It doesn't really matter. So that's the phenotypic ratio. That's the expected phenotypic ratio. 9 out of the 16 will be purple and smooth. We don't care whether they're homozygous dominant or heterozygotes. Okay. 1, 2, and 3 of them will be purple and wrinkly. Okay, that's this one down here. 3 out of the 16 are going to be purple and wrinkly. 1, 2, and 3 are going to be yellow and smooth. And only 1 is going to be yellow and wrinkly. So we have this phenotypic ratio of 9 to 3 to 3 to 1. Dihybrid crosses equals 9 to 3 to 3 to 1 ratios, expected phenotypic ratios. So remember, dihybrid crosses where both parents are heteros for 2 traits will always produce a 9 to 3 to 3 to 1 ratio in terms of the distribution of the phenotypic conditions amongst the offspring. All right, so this is kind of what it would look like. In the lab manual asks you to take a look at a corn cob like this and count the kernels, how many of them for each row, how many of them are going to be purple, smooth, purple, wrinkly, yellow, smooth, yellow, wrinkly. All right, so I didn't bother counting this picture. I had my own here where I was preparing this lab, and I got these numbers. You can copy these numbers, or you can go ahead and try to count these corn kernels, just 3 rows if you want, if you'd like. All right, and in this case, in row number 1, I have 10 purple smooth, 5 purple rough, 2 yellow smooth, 2 yellow rough. Okay, so that gives me a really weird phenotypic ratio of 5. All right, to what is it? 2.5 to 1 to 1. Right, way off, way off. And the other rows are kind of the same too. But when I add up my samples and I get a larger sample size, I get something much better. 8.7 to 4 to 3.7 to 1 ratio. Very much closer to my 9 to 3 to 3 to 1. Right? Remember, we're comparing it to 9 to 3 to 3 to 1. So my expected compares relatively well to my, just by eyeballing, to my actual phenotypic ratios. Okay, I'm going to share my screen one more time. I think that will be it for the PowerPoint, actually. Okay. So here they're discussing how you distribute the alleles and use the FOIL method. Remember back to your algebra. Anyway, all right. So here it says, obtain an ear of corn. Supply table, you will be examining the results of the cross, this dihybrid cross. Do the Punnett square do the Punnett square on page 91 in order to state the expected phenotypic ratio among the offspring. So that would be 9 to 3 to 3 to 1. That's the Punnett square that I presented to you on the PowerPoint. I think that's it. So you guys can fill this out from the PowerPoint. Okay. And also, you guys can do this from the PowerPoint, right? Now, calculate the actual phenotypic ratio based on the data and record them. Do the results differ from the expected phenotypic ratios? Yes. Per class data, slightly, but yes. Remember, larger sample sizes, this is size, larger sample sizes approach expected ratios. All right. Okay. We're going to skip this wing length of Drosophila. We never do this in lab anyway. And of course, we're going to skip the parts that result from that. The next thing we're going to discuss are these X-linked crosses. All right. So remember that you have most of your chromosomes are going to be called autosomal. You have 22 of those, 22 pairs. And the sex chromosomes, you have one pair, right? So you have 23 pairs of chromosomes altogether, and one pair are sex chromosomes. That's the X or the Y chromosome. And the rest are autosomal chromosomes. They have nothing to do with sexual expression. Right. X-linked crosses. X-linked crosses are all about the sex chromosomes, specifically the X chromosome. All right, so remember, well, actually not remember, but note that differing sex chromosomes don't occur in all animals. It occurs in many animals, though, but not all, or for that matter, organisms. For example, doesn't really happen in plants. Okay, all right, so like I said before, the autosomes are most of your chromosomes that have nothing to do with the expression of your particular sex. And then you have the sex chromosomes, the X and Y. All right, if you're a female, you have 2 copies of the X chromosome, and if you're a male, you have 1 copy of the X chromosome and 1 copy of the Y chromosome. All right, some alleles on the X chromosome have nothing to do with gender. That's true. And so these genes, these particular genes, are said to be X-linked. So they're found on the X chromosome, though they may have nothing to do with whether you're a female or not. The Y chromosome does not carry these genes. In fact, the Y chromosome is all about being a male. Okay, and so, well, let me backtrack. The Y chromosome carries different genes than X chromosomes. So, X and here's something for you guys to write down. X and Y chromosomes are not homologous. Okay, they do not pair up in meiosis. You do not want your X chromosome exchanging genetic material with your Y chromosome. They do not pair up. They do not have or carry the same types of genetics or genes, let alone the same traits. Therefore, males don't have a backup to the X chromosome or to the genes on the X chromosome. So whatever genes are on the X chromosome for males, that's what their phenotype is going to be. The genotype is going to equal their phenotype in terms of excellent traits. Right? So for males, X-linked traits, the genotype is the same as the phenotype. For the female, because the females have 2 copies of the X chromosome, right? The X chromosome will have its homologous. All women will have 2 homologous X chromosomes. They have 2 copies of each trait on the X chromosome. So the regular rules apply. You need 2 copies of the recessive allele for it to be expressed in the phenotype. Okay, so in this example, in fruit flies, red eyes are dominant, which is an excellent trait. So we write X for X chromosome and the little r for red eye trait are dominant over white eyes. So X chromosome with a little r. You will be examining the results of the cross, this cross here. Okay. Complete the Punnett square and state the expected phenotypic ratios. All right. So in this case, let's do the sperm, the male, right there. X, big R, and Y. And for the female, big X, right, big X, big R, and little r. So what are going to be the expected ratios of the offspring? Okay, we have a female there being born or being created. If this egg is fertilized by that sperm, you have a female. And both are red-eyed females. In this other case here, another female, but it's going to be heterozygous for the red eye condition. However, red eye is dominant, therefore its phenotype is red eye. This will be the row for the males. Okay, and in this case, this is a recessive and this is a dominant. All right. So how many females will have an expected phenotypic ratio? What is the phenotypic ratio for the females? The phenotypic ratio is 1, just 1. All, or you can say 100%. 100% red eyes. The males, half, 1 to 1, okay, or half will be red-eyed. So with this example, I hope that you guys have a good idea of how X-linked chromosomes, traits, X-linked traits work. It's the same rules that apply to the other traits that we were discussing today, except that it's linked to either the X chromosome. It's linked to the X chromosome, right? They do have Y-linked chromosome, Y-linked traits, but they're going to be quite different, right? Different from what you find on the chromosome, because once again, the X and the Y chromosomes are not homologous chromosomes. All right, so in this case, we're going to go ahead and skip these portions of the lab, and we're going to this will take us to the review questions. Okay, what are the alternate forms of a gene called? And that is allele. Okay, and again, these are the types of questions that will show up in tomorrow's lab exam. What word is used to describe the possession of one dominant allele and one recessive allele? What word is used to describe the possession of one dominant allele and one recessive? So each of the alleles is different. Right? We're talking about heterozygous. What word is used to refer to an individual's appearance or behavior? Or the phenotype. Which of Mendel's laws says alleles segregate during gamete formation so that each gamete contains only one allele for each trait? Okay. So we go back and we read. Each organism contains two alleles for each trait, and the alleles segregate during the formation of gametes. Each gamete then contains only one allele for each trait. Right? That's the law of segregation. Okay, 1 second, guys. Okay, sorry about that noise. What is used to determine the results of a cross between individuals with known genotypes? What is used to determine the results of a cross between individuals with known genotypes? We use a Punnett square, 2 n's, 2 t's. I don't fit. It doesn't fit there. Let me just draw a square. Punnett square. If the phenotypic ratio among the offspring is 3 to 1, what is the genotype of the parents? Well, both parents are heterozygous. They're both heterozygotes, whatever you want to say. Heterozygous. How would a tobacco seedling with the genotype little c for chlorophyll be described? So that would be right, homozygous, recessive. Check my spelling. If the allele for a purple corn kernel, P, dominant, big P is dominant over the allele of a yellow corn kernel, what is the genotype of the yellow corn kernel? Well, the genotype is going to be little p, right? Or homozygous recessive. Okay. What is the cross between two individuals with this genotype called? Right? Both traits are heterozygous. We call that dihybrid cross. How many of the offspring of the cross between two of these individuals, the same, will show the recessive phenotype? Right? Remember, the ratio is going to be 9 to 3 to 3 to 1. Only one. Or you can say 1 out of 16. One in 16 will show the recessive trait. Which of Mendel's laws says the separation of one pair of alleles into gametes does not impact the separation of a second pair of alleles. It's a law of independent assortment. Okay. And how many different kinds of gametes can be produced by a parent with this genotype? Well, you can figure that out, right? One gamete will be big G with little r, and the other one will be a little g with little r. So only two. There's no other possible combinations. What is the name given to the chromosomes that do not actively determine the gender of an organism? All right, these are autosomal. Autosomes or autosomal. Can a female fruit fly that is homozygous dominant for red eyes, have any offspring with a male with white eyes? All right. Have any offspring, male or female, with white eyes? Well, you can do a Punnett square just to make sure, but it doesn't look like it. No. All right, male is going to be, let's say, has white eyes. All right, let's give him the best chance possible. But in any case, the phenotypic ratios are going to be, the phenotypic expression is going to be normal, the dominant. I apologize, dominant. So, right? Remember, the X chromosome that males inherit only come from the mother. And if the mother is normal, then the male offspring are also normal. And if the mother is normal, even if the father, I keep saying normal, I'm sorry. If the mother is dominant and only dominant, then even if the father has the recessive gene, then because the female offspring have two copies of the X chromosome, they will also be phenotypically dominant. So can the female fruit fly that is homozygous dominant for red eyes have any offspring with white eyes? The answer is no. Okay. All right, we can skip this. And we're going to head off to chapter 11, which, as you can see, we're going to skip quite a few things in chapter 11 too. All right. So in chapter 11, it's about the same topic and actually doing some genetic investigations on based on what you learned in the last chapter. Okay. So, what happened there? Okay. So in this case, we have a few examples of autosomal dominant traits and recessive traits. What is the homozygous dominant genotype for type of hairline? Okay. So here they're showing that here. Widow's peak happens to be a dominant condition. So big W, big W. What is the phenotype? Widow's peak. A widow's peak is present. Widow's peak is just this hairline, like Dracula, right? All right, and having a straight hairline means you have the recessive condition. What is the homozygous recessive genotype for finger length? Here's some. Either you are a type of person that has short fingers, which happens to be the dominant condition, or long fingers, which happens to be the recessive condition. So you have little s, little s. What is the phenotype? Long fingers. The phenotype for the recessive genotype. Why does the heterozygous individual that has big F, little f have freckles? Okay, that's this. Okay. All right. So because freckles are dominant. All right. According to this, if you have freckles, you have the dominant genotype. You have the dominant allele in your genotype. Either two copies or you're heterozygous. If you don't have freckles, then you have the recessive gene. Okay, so here are some questions. Maria and the members of her immediate family have attached earlobes, so here we're talking about this. What is Maria's genotype? Okay, Maria and the members of her immediate family, it's her mom or dad and her children, if she has any siblings also, have attached earlobes. That's this. Therefore, if you have attached earlobes, you have this genotype. Her maternal grandfather has unattached earlobes. Deduce the genotype of her maternal grandfather. All right, so her maternal grandfather was either homozygous dominant or heterozygous. Okay, he had to have at least one copy of the dominant allele. All right, so her maternal grandfather has unattached earlobes, but her, I guess, yeah, but her mom, right? Her mom has the recessive condition. Right? So her mom has the recessive condition. It's possible that you had big E. It's possible that the mother would have been little e, right? But the grandfather have been this. No, right? So it pays to do the Punnett square. So the grandfather has to be a heterozygous. Can't be homozygous, right? Because we know that her mom, is recessive. She's recessive. Her mom is recessive, and her mom's father had the dominant condition. But the only way that her mom could have been recessive is if both her grandmother, maternal grandmother, and maternal grandfather had the recessive allele. All right. Okay. So number 5, Moses does not have a bent little finger, but his parents do. Deduce the genotype of his parents. Bent little finger. All right. So the bent little finger is this thing here. Okay, the little finger bends toward if it has a bent little finger, it's dominant, and if it's straight, it's recessive. Moses does not have a bent little finger. That means he's recessive. But his parents do. That means they're both heterozygous. Right? They both have to be heterozygous for the same reasons that number 4 and her grandfather had to be heterozygous. Okay, so big L, little l for both. Let's see. word they're looking for is both of Moses' parents are big L, little l. Make it fit. Answer. Why do both have to be big L? Because it would be a dihybrid cross, right? Sorry, not monohybrid. Monohybrid crosses will produce recessive offspring. All right. He is recessive. Both his parents are not. So we know that both of his parents have to carry the recessive allele in order for him to have inherited 2 copies of the recessive allele. You can say that instead of what I wrote. All right, Manny is adopted. Okay, Manny is adopted. He had hair on the back of his hand. All right, so that's one of those traits over here. Hair on the back of your hand. If it's present, it's dominant. If it's absent, it's recessive. Manny is adopted. He has hair on the back of his hand. Could both of his parents have had hair on the back of the hand? Okay. We're not sure. You know what? Let's cross this one out because what are they talking about? Are they talking about adopted parents or biological? If they're talking about biological, then why include this term, adopted? So I don't like this question. Goodbye. It's not possible. We can assume that we're talking about adopted, in which case it wouldn't matter. All right. All right. So you guys in lab, we would have been comparing one another, ourselves to each other, and seeing who's got the widow's peak, who's got attached or unattached earlobes, and freckles, and hairy hands, and does your thumb bend backwards or not? And do you have bent little fingers that bend towards the rest of your fingers? And interestingly enough, interlacing your fingers. When you cross your fingers, cross your hands, right? Put your fingers, interlace them with each other. Which thumb lies comfortably for you? Which thumb lies comfortably for you on top of the other thumb? Is it going to be the right or the left thumb? All right, so we don't have to do this though, but you guys can read through this and see what you have. All right. Remember, hitchhiker's thumb is something that's really bent backwards. Determining inheritance. Recall that the Punnett square is a means to determine the genetic inheritance of offspring if the genotypes of both parents are known. In a Punnett square, all possible types of sperm are lined up vertically and all possible types of eggs are lined up horizontally, or vice versa, doesn't really matter. So that every possible combination of gametes occurs within the square. Okay. So, for example, with the widow's peak, right? This female is recessive, so she only has one possible type of allele. There's no reason to have to do it again. It would be the same thing, right? It would once again be the same thing. Okay, and for the male, this male is heterozygous, so he has 2 possible alleles. So sperms can either have the dominant allele or the recessive allele. So the Punnett square, this side of the Punnett square is going to be the same as the one that's shown. Right, so big W, little w, big W, and little w. Sorry, I apologize for that. Wasn't paying attention. little w, little w. Okay, so exactly the same. Okay, in figure 11.4a, let's see, where is the little figure? Ah, here we go, 11.4a. Quarter of the offspring have the recessive phenotype. That's this one. What percentage? You have a 1 in 4, right? 25% chance. 3/4 of the offspring have the dominant phenotype. That's the remainder, 75% chance. In the next one, 11.4b, this one, okay, that's half and half. Half of the offspring have the recessive or the dominant phenotype, so you have 50% chance. With reference to these figures, 11.4a, if a genetic disorder is recessive and both parents are heterozygous, so these A's that they drew here mean for the time being until they tell you what they mean. With reference, if the genetic disorder is recessive and both parents are heterozygous, which they are, okay, big A, little a, what are the chances that an offspring will have a disorder? So it's 25%, right? A 3-to-1. This is a monohybrid cross. So you have a 3-to-1 ratio. With reference to figure 11.4, if the genetic disorder is dominant and both parents are heterozygous, what is so that's big A, little a. Both parents are heterozygous. What are the chances that an offspring will have the disorder? And we see, sorry for moving it so much. Both, wait a minute, why are they saying 11.4b? Oh, 11.4a, I apologize. It's this one again. This one again. What are the chances, if it was dominant, what are the chances that an offspring will have the disorder? Well, you have 75% chance, right? Now, for figure 11.4b, if parents are heterozygous, big A, little a, by homozygous, little a, little I, homozygous recessive, that is, and the genetic disorder is recessive, what are the chances that the offspring will have the disorder? So let's take a look. All right, here one of the parents is recessive and the other parent is heterozygous. What are the chances that you're going to be the recessive individual? And that's 50%. With reference to the same one, if the parents are heterozygous, big A, little a, by homozygous, recessive, little a, and the genetic disorder is dominant, what are the chances that an offspring will have the disorder? This is going to be the same thing, 50%. Okay, so you see, 50% of the offspring have one phenotype, and 50% of the offspring have the other phenotype. The phenotypic ratio is 1-to-1. Okay, so this is an interesting read, but we're not going to need this for this lab. We don't need this. All right. And here to discuss X-linked disorders, it's virtually the same thing as the X-linked trait with the fruit flies in the last chapter. Here's a good question. Does a colorblind male give his son a recessive bearing X or a Y that is blank for the recessive allele? Okay. So, we're talking about X-linked recessive disorders. Only females, right? So the answer is no. Only mothers can pass down that trait to their sons. Why? Because males do not give their sons an X chromosome. They only give their sons Y chromosomes. All right, so here is a key, our key that we're going to use. All right, you can be homozygous dominant for normal vision, heterozygous condition for normal vision. And for males, of course, if you inherit one of the normal X chromosomes, you have normal vision. On the other hand, if you have if you're homozygous recessive for this color vision in the female, then you're colorblind. And for males, all it takes is one copy of that recessive X-linked trait. Remember, you have to inherit that from your mother, otherwise you can't. Otherwise, you can't get that from your father. All right. So an X-linked recessive disorder in a male is always inherited from his mother. Most likely, the mother is heterozygous and therefore does not show the disorder. She is designated a carrier. Right. So, for example, these are the carriers. Figure 11.5. Where is that? Here we go. Shows how females can become carriers. Use the key to genotypes and phenotypes for X-linked recessive disorders to complete the parentheses of questions 2A with genotypes of the parents. What is a genotype of a colorblind female? Colorblind female would have to be X little b, X little b, right? How many recessive alleles does a female inherit? To be colorblind, she must inherit 2 copies. What is the genotype of a colorblind male? X little b and that Y chromosome to make a male. How many recessive alleles does a male inherit to be colorblind? Just 1. Tough luck, right? So sorry about that. Okay, that's the problem with being a male. It really does affect how long we live, and other things as well. Just having 1 copy of the X chromosome. Right? That's why I say tough luck. If there are studies that show that have looked at the effects of longevity, how long something lives, in species that have males or females with different sex chromosomes. Most species that do have sex chromosomes that are different, the males are the ones that carry the 2 different chromosomes. And on average, statistically significantly, the males live less time, have shorter lifespans. Tough luck, right? And in those, and just to show that it's not just about being a male, some of the species that they studied, which have females where the sex chromosomes are different, the females in those species are the ones that live less time. All right, so yeah, tough luck for us, guys. Oh. All right, with reference to figure 11.5a, that's this, right? Yeah. If the mother is a carrier, right, so that means she's like this, and the father has normal vision, what are the chances that a daughter will be colorblind? Chances, right? Because, all right, here are the daughters. A daughter will always inherit a dominant allele from one of the parents, or both. 0. A daughter will be a carrier. What are the chances that a daughter will be a carrier? 50%. Okay, take a look. One of the daughters is normal, and one of the daughters has. I apologize. Oh, wait a minute. I thought the father was normal. Again, I'm looking at the wrong one. No, I'm not. It says 11.5a. All right, so actually this is wrong here. All right, so we're actually looking at this one, where the mother is a carrier and the father is normal. Am I correct here? Where the mother is a carrier and the father has normal vision. Mother is a carrier and father is normal. All right, so they made a mistake here. So this is actually B, 11.5B. If the mother is a carrier, X like this, and the father is normal, all right, and so half, let me erase this here, half of the, what's the question? I forgot now. A daughter, what are the chances a daughter will be a carrier? Okay, yeah. Here are the daughters, and only one of them out of the two will be carrying the recessive allele. What are the chances a son will be colorblind? Okay, that's 50%. With reference to 11.5b, if the mother has normal vision, okay, so now we know this is actually this one. That's 11.5a. They made that mistake. Okay. If the mother has normal vision and the father is colorblind, right? This one. What are the chances that the daughter will be colorblind? 50%, right? So here are the daughters. If the mother is totally normal, then actually yes, there's zero chances because the daughter will be a carrier. A daughter will. What are the chances the daughter will be a carrier? This is 50%. All right. The worst case scenario, daughter will be a carrier. I'm sorry, a daughter will be carrier 100% of the time, like I said before. I apologize. I don't know why I'm second guessing myself here. 100%. Okay. What are the chances that a son will be colorblind? 0. Right, because it's the father that is the colorblind one, and fathers don't pass on their X chromosomes to their sons. Okay, this is just a repetition of the same thing. We don't have to do that again. Okay. Blood types. Blood types are a special case. So the A blood type and the B blood type, the alleles that code for that are both dominant. All right, so that's a weird case where you have multiple dominant alleles. And in the population, you can have either one of the two alleles or both, or the absence of either one, which is the O blood type. All right, so we have a situation where we have codominance. A man with a type A blood reproduces with a woman who has a type B blood. Their child has blood type O. Why? Okay, so we'll do a Punnett square. A child has a blood type O, which actually means he's got missing both alleles in both chromosomes. Okay, and the mother. a man with a blood type A, so he could be A, O, and the mother can be B, O. So one of the offspring, there's a 25% chance that the offspring will be AB blood type, or just B blood type, or just A blood type, and 25% chance it will be O blood type. The man will be AO, the mother will be BO, and the child will be OO. The only way that you don't have A or B is if you have the O blood type. If you're recessive for both, for having O blood type. Okay. So it's not really a recessive condition, it's more of an absence of the presence of the B allele or the A allele. If a child has a type AB blood, and the father has a type B blood type, what could the genotype of the mother be? So let's pretend this is the father here, and this is the mother, right? We already have the Punnett square for that. So the mother would be AO. Okay, so the mother would be AO, or could the mother be AA? AB, AB, yeah. Mother could be AA. If both mother and father have blood type AB, both are AB. If both mother and father have blood type AB, they cannot be parents of a child who has what blood type? Okay, what's not possible? Well, A blood type is possible, AB blood type is possible twice, and B blood type is possible. What's not possible? O is not possible. All right. And then here they're asking you for what are the possibilities. Again, we have one parent is AO and the other is BO. And you have AB, and here you have just B, here you have just A, and here you have just O. Okay. 1 second, guys. Okay. Apologize. All right. We're going to skip the rest, basically the rest. No, we're not going to do this. We don't need to do that. All right. And we're not doing that. And that takes us to the end. All right. What phrase describes an individual with two dominant alleles? Two dominant alleles. That's homozygous. Homozygous dominant. What word refers to the alleles possessed by an individual? We're talking about what is the genotype. How many autosomes do humans have? 22. An individual will have a widow's peak if the genotype big W to big W or big W little w is present. Is widow's peak an autosomal dominant or autosomal recessive? Autosomal dominant. What technique can be used to determine the genetic inheritance of offspring when parents have known genotypes? What technique can be used to determine the genetic inheritance of offspring when both parents have known genotypes? Use the Punnett square again. I feel like we got that question asked before. Punnett square. Cystic fibrosis is an autosomal recessive disease. What word refers to an individual who has one recessive allele for cystic fibrosis? If it only has one recessive allele, then we're talking about a heterozygous. Heterozygous. Okay. Or carrier. Maybe they're looking for carrier. Okay. From whom does a male child inherit color blindness? It would be the mother. Right? I explained that a few times, right? The color blindness gene trait, the allele for that is found on the X chromosome. And since only mothers give their X chromosomes to their sons don't receive it from their fathers. How many dominant alleles does someone with AB blood type have? 2. Can a child with AB blood type have a parent with O blood type? Yes, right? The parent can be AO or BO, right? Or both. What is the presence of 3 chromosomes instead of 2 called, and what human chromosome is most commonly affected? All right, so there they're talking about a condition known as trisomy, the most common being trisomy 21, which causes Down syndrome. Yeah. 21. What is created when chromosomes are paired by size and shape? All right, so when you take the chromosomes out of the cell and you do a little study and assay, you want to see what the chromosomes are, and you line them up nicely. So this is how they figured out how many chromosomes humans have, and other animals do, or organisms. You call that a karyotype. Karyotype. Okay, what is the actual chromosomal makeup of an individual? So that's what this is. These are the karyotypes. Okay, and by the way, this one shows that trisomy 21, 3 copies of the 21st chromosome. Okay, so we're going to skip this. All right, so that will be it for today. All right, guys. So tomorrow, I sent out an email earlier today describing what you should expect for tomorrow with the exam. The exam is on Blackboard, and the exam will appear under content in the Blackboard page. The exam will open at 9 a.m. and it will close at 7. So make sure that it's only 25 questions. It shouldn't take you longer than 10 minutes, maybe 25 minutes to complete it, but I'm giving you a maximum amount of 1 hour to do so. However, the exam closes at 7. So if you start the exam at 6:50, you only have 10 minutes. All right, that should not be an issue for anyone because you all signed up for a 4 o'clock lab or 9 a.m. lab. So technically, you should be available at those times. And therefore, you should have plenty of time to take your exam. All right. Okay. So, however, if you guys have any issues with connectivity or being able to take the exam online like that, just let me know, and we'll figure something out. And make sure you read that email that I sent out to the class earlier today, and that should give you information on that. More information on the exam. Okay. All right. Study up. The exam questions, again, they're going to be based off the short answer questions at the end of the chapters. Most of them will be fill-ins, and some of them, I think there's only 3 that will be multiple choice. Make sure, you guys are home and you have your books. So make sure that you spell everything correctly, because what happens is with the system of fill-ins on Blackboard exams, if you spell it incorrectly, it just doesn't recognize the word that you wrote, and it automatically marks it as wrong. And since you guys have the books, you don't have an excuse, so I'm not going to bother fixing it. On the other hand, if you have a correct answer, you manage to figure out a way to say the correct answer in a way that I couldn't guess, but nevertheless your answer is correct, of course I will give you the credit for that. All right. So you will get a grade at the end of the exam, but that is not going to be, doesn't have to be the final grade for your exam, especially if you have a grievance with your grade. We'll look through it and we'll see what it turns out to be. All right. All right. Enjoy the rest of your night and rest up for tomorrow. Stay home. Stay safe. Not stay home. Stay safe and stay healthy.